Good afternoon, sir.
So as I am going to start the new topic from second unit, that is the good laboratory practice. Till now we discussed the good manufacturing practice, then good agriculture practice, good agriculture and collection practice. Now this is the time for GLP, right? So as the GLP may be defined as a body rules, operating procedures and practices established by a given organization that are considered to be mandatory with a view to ensuring quality and correctness in the results produced by a laboratory. So, as the name indicate, good laboratory practice. So, till now, when we were discussing about good manufacturing practice or good agriculture practice, this is very much clear after completing this many class in this good word. So, whenever the good word it is coming, we are concerned about how best we are going to perform the methodology or the procedure somewhere so that we can completely concern with the quality of the outcome, whatever it may be. Whether it is dosage form or crop or medicinal plant or raw material or finished product, we are expecting the high quality product. So for that high quality product, for that high quality outcome, we need to adopt some types of good tactics or good technology so that we are able to achieve that. Or another definition you can see here, the GLP is a quality system concerned with the organizational process and conditions under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, achieved, and reported. Right. So it is not that simply you are performing and completing the task. We have to plan it, that what we are going to do in the lab, how we are going to do in the lab, and how we are going to perform that particular method, how we are during the performance, how we are going to manage or monitor the different departments or different area of this that procedure, and everything we need to record right from the beginning to the end, and it has to be saved or achieved, and later on whenever required it will be able to report to different bodies and different persons, right? So when we look at the history of the good laboratory practice, let us see that what happened actually, and when it started. The GLP was first introduced in New Zealand and Denmark in 1972. So these kind of questions you may get in your objective type of question, right? When this good laboratory practice was introduced, in which country or in which year, like that, right? So these kind of things you need to remember or you need to take care about. So here GLP was first introduced. So when it was introduced, when GLP was first introduced, question may come, in New Zealand and Denmark in 1972, and later in US in 1978, in response to the industrial biotest labs scandal, right? So this already we have seen that how it happened. So after that scandal they started doing this. It was followed a few years later by the Organization for Economic Cooperation and Development, that is called the OECD guidelines, principle in GLP in 1992. The OECD has since helped promulgate GLP to many countries. So they already started. Then after that, this organization that is called OECD, that Organization for Economical Cooperation and Development, to the different countries throughout the world. So here you can see that different elements of GLP, that is the certification of laboratory facilities, certification of analyst, specimen or sample tracking, documentation and its record, accountability, reagent and materials certifications, statistical procedures, instrumentation validation, and quality assurance. Standard operating procedure for GLP usually provide an elaborate description of various ongoing activities carried out analytical laboratories. The examples are sample handling and preparation, analytical method used, proper maintenance of all instruments, and record keeping. Quality assurance unit. So when it comes to the quality assurance unit, is entirely responsible for assuring good laboratory practice being implemented effectively, right? So what do you mean by effectively? Means when we are maintaining or implementing the such type of records in the GLP, it has to be implemented properly. That's what. Then coming to the next one, the prime responsibilities. The prime responsibilities of that are exclusively fall within the domain of quality assurance unit are: the first one, implementation of quality procedure. Procedures they will make, procedures will be available. Procedures are what do you call, uh, for anything, for let it be for the making biryani, procedures are there. But if you are preparing in your own house or somewhere, you need not to follow the procedure, you need not to implement the Hyderabad house procedure or the paradise procedure or the any reputed restaurant procedure which you know or which is available into the public domain. It is not necessary that you need to implement. But here, when it comes to the laboratory practice and the quality assurance unit, they have to have the responsibility, prime responsibility, that they are going to implement the quality procedure which has been authenticated, which has been recognized. That procedure they need to implement. The second one suggests that assessment of quality procedures on a continual basis. It is not that the one day if inspecting teams are visiting to your industry, you have adopted that and you have completed the you have taken the quality procedure and you have done that day task only. It should not like that. It should be the uniform or continual basis, every day, every month, every year, the same procedure, until and unless it is not being updated, it has to go on. It's not that the any inspecting body or governing body they are coming to the industry or unit or laboratory, that day only you are making clean sanitized, and every machines are being cleaned by the people who are employed there. And one day, if it is like how it happened when the inspection comes, right? So everything looks like oh, it's very great, everything is going proper. One day only, so very next day everything will be not be there, right? Even number of staff, number of machines, number of the different things, so that are going to change. So it should not be like that. So it has to be in a proper way. Audits of the laboratory periodically, right? The audits that we need to audit, the inspecting team need to audit the lab, right? There will be the sequential audit. One audit will be monthly basis, one audit will be 6 month basis, then yearly basis. So like that, you need to audit the lab in periodically, right? So that's what the routine checkup, I mean to say is, routine checkup is very much essential. Objectives of good laboratory practice: it is the first one, adopt good and safe operating procedures and recording systems. It should not be that You adopted some other types of operating procedure which is not suitable for the machineries, which is not suitable for the finished product. It should be good and safe. The procedure which you are going to implement, that should be one good. Good means quality wise, good means utilized by it, good means in all aspects, and it should be safe also for everyone, the ultimate consumer of the your products, the people involving into the procedure, everybody, for it should be safe. And a recording system, the thing which we are doing in the lab, that should be recorded, documented properly. The second one suggests in objective that prevent human errors in the performance of the job. That what may be the human, what do you mean by human error? Human error means sometimes what happen, if you'll forget to switch on the fan, but fan is required over there because ventilation is very much important to dry your product, to dry your raw material, to dry your liquids. And if you are forget, if you are the because of xyz reason, maybe the age, maybe the reason, the health is not proper or some other reason, but so that because we are not the machine, we are not going to follow the commands in 100%. So if so that's what we need to minimize that, we need to prevent that, that kind of what you call forgetting the things, and the common error which is possible with that job. Okay, the third one, the objective third one suggests that prevent equipment errors in measurements, right? So equipments, they are machine. Human body is also machine. The human body also can make the mistake, the machine also can make the mistake. So what are the type of errors? It may be, suppose if you take the small vernier calipers or screw gauge, that we have studied in our schools, that there may be the positive error, there may be the negative error. So the error we are finding out there. So likewise in different machines, when they are in use for the long time, they are into the employed for the long time, the machines are going on. So slowly they will be with the lot of using the small small gaps will come, small errors will start doing. So that's what we need to have the proper record or we have to find out that error so that it will not going to interfere our products. The fourth one suggests that prevent unsafe and hazardous acts which could affect individuals and earn or properly. It means unsafe and hazardous act means, for example, you are dealing with gas, you are dealing with some chemicals which is hazardous chemical. For example, strong acid or alcohol or benzene or ether or petroleum or highly inflammable, what do you call, liquid gas. If you are dealing with those kinds of things, you have to be very very careful. Right from the beginning, your shoe, your dress, your hand gloves, your mask, your how these bum squad people, they will wear the dress. So where you are working and how you are working and what for you are working, you should be very much aware when you are working into the lab. And these kinds of things, if you will not take care, the fire accident will takes place. You might be seeing that in this laboratory that fire came and this many people died, and this much crores rupees that has gone into the water, and that company was not undergone for insurance for half of its property, which is not refundable. Forget about the employee, those who are working there, and they are victim of the that particular types of incident. But what about because any industry, any laboratory, it is not one person property. It is the society. It is many people, shareholders, their staff, their what do you call, labors, right from the each and every person, those who are getting benefit from that particular organization, everybody will go into the loss. They may lose their life, they may lose their job, and they may lose their future. right? So that's what we have to, that is what otherwise what is there in the lab, what is happening, because the good laboratory practice, they it is helping to prevent the unsafe and hazardous act, because when we start doing the good, when we start implementing the good method or good procedure, automatically it will come into the habit, and by seeing us, the other company, the small laboratory also, they will start implementing such kind of good procedure or good method, then it will go on. Improve efficient performance of the job. Efficient performance means, what do you mean? That's what there is always value for the experience, there is always value for the leadership. Why the leader should be proper, why the experienced person only should be the leader, why it is necessary. Immediately after joining into the field, anybody cannot become leader. That's what efficient performance. When you're dealing with the critical job, when you're dealing with the critical types of procedure, you need to have experience, and experience will help you and your team to complete the task without error or less error. I cannot say 100% perfect you are going to perform that one, but you can minimize the error, you can minimize the hazardous act, you can minimize the unexpected incident when you are having the good team, when you are having the good leader, when you are having the experienced performer. So these are the main, very basic objective that is behind this. So here you can see this triangle shape of the graph, the successful, how it is, how or when it is going to be successful. The veterinary staff, study directors, quality assurance team, regulatory team, and a contributing scientist. So when I say the laboratory, generally your college lab will come in your mind, right? That is a common phenomena. Or you may go up to the Niper lab because we visited CCMB labs or something like that. So it is lab means it is not that the only your faculty will be there and one lab technician and one lab assistant, few microscope, few test tube, few glassware, few wash basin, two or three null in working condition, one hot air one. That is not lab. Let me tell you, that is not a lab. When I say lab, you can see in your graph here, veterinary staff. Veterinary means the doctors or the physicians for the animals. Study directors. Study directors means these are director also having the high qualification on their respective field, and they are only going to design that what on what topic, on what subject, and what problem the laboratory is going to have their observation or study. The quality assurance team, this team work only to look after the quality of, from the raw material, from the initial product to till the end of the study, right? It may be finished product, it may be tablet, capsule, ointment, lotion, anything is you are going to be, or it if you are going to have the result, any drug or whatever it may be. Regulatory team, it is going to control the rules, regulations, the procedures, methodology, tactics, everything. Contributing scientists, because scientists are the major player here, and they are going to design different projects, duration of the projects, the cost of the projects, the how much staff required, scientists required, everything they will be planning. So if the idea behind GLP, what an idea, Sarji, right? So it belongs to the functions of the respective authorities to make sure that the safety and the environment is proven, safety claims can be verified, right? So what is the functions of the respective authorities. Safety and safety claim, it here you can see these two words, it is being highlighted here, right? So your safety is our concern. That will be many company, they will be giving their quotes, right? So since it is laboratory and we are dealing with the different hazardous machineries and chemicals, ultimately we are human being, and those chemicals, those machines can harm us. That is for sure. Lab means second things comes hazards. Risk is there. Life risk is also there. Gas, petrol, fire, anything it may happen. Even in pharmacognosy lab, also smallest lab in the world, you can say, if you think about there also there are a lot of chances of to being injured. And when it comes to the environment, because yesterday I was gone through one movie in Hindi that is called Irada. One movie is there, Nasruddin Shah and Asad Warshi. That movie is completely based on this only, GLP only. And luckily I am dealing with the GLP today, right? So many of the industries or for companies, from not only pharmaceutical companies, chem field is there, relief is there, leather industries are there, the different types of meal, paper mills are there. What they're doing, whatever the toxic chemicals or toxic outlet it is coming from their industry, they are making hole into the land, and every drainage, every wastage of their industry, they are putting into the land inside, maybe half kilometer, maybe 50 feet, 100 feet, 200 feet, 300 feet, depend on their wastage quantity or wastage volume. So what is happening? When they will go inside, they will start dispersing as per the space available there, and they are not going to change their form. They will be as it is, and slowly they will dissolve in first into the soil, then later into the water, and ultimately it will reach to the surface once again. And it is, these chemicals are carcinogenic, hazardous. If you touch this chemical also, you may get cancer. Need not to swell or need not to inhale. Touching itself, if it will mix into the water, suppose in that area there is a canal or lake or any kinds of water reservoir, dams are there, people are using those that water for bathing purpose, for drinking water purpose, for so many other uses. What will happen if this wastage chemical linked with that water, because you are disposing them inside the soil, but what is happening, you have not fixed the microscope or some camera over there that you are looking and observing what is happening. And it happened in that movie also, it happened, and you can see in this lot of newspapers are available, lot of news happened. That Bhopal gas plant is also is one of the greatest example, which will be never be forgettable. The many people they have died, like more than the coronavirus, they have died over there. And the fraction of the government also were not able to in the not was there in that position that it can control the people's dying, because it was gas leakage. When such kinds of hazardous gas, such kind of toxic gas will release into the air, how you will save the people will die in front of the you will be also dying, though nobody can help each other. That's what safety measures is very very important. Second thing, in idea behind the GLP, that A regulatory authority may then obtain verification of the such an experimental result by either two-way itself or step by step. The GLP principles are designed as a tool enabling also the improvement of study and data quality. So it is not only for the safety purpose. Here you can see that it is designed a tool which will be enabling or which will be helping so improvement of study and data quality. Data quality in the sense each and every data in the outline process, inline process, and depth process right from the beginning. What is the major data you are supposed to compile? For example, you are working in let me tell the example of LCMS, liquid chromatography and mass spectroscopy. So what is the type of chromatogram, what is the type of solvent, what is the type of mass spectroscopy, the bombardment size of the, so which sample you loaded, how much electron bombardment is there, and how much elute you have got that. So when you have involved into the procedure, you need to collect the quality data which will help you in future and during the study both. All these requirements can be summarized in three issues that are central to the ideas behind GAP. The first one, possibility, accountability, and GLP increases awareness. So let us understand what is the possibility behind this. To reconstruct the whole course of a safety study, even years after it has been performed, and even in the absence of persons having been actively involved in the conduct of this specific study. Okay, that's what reconstruct the whole course of safety design, and even years after also the it has been performed, and even the absence of persons having have been actively involved in the conduct of this specific study. Example, all the animals have received the correct dose of the test item at all time, that the correct sample have been taken and analyzed, and then actual data that has been collected. So that is called these reports submitted to the regulatory authority. So we have to check the possibility, and that has to, for the this purpose, the regulatory purpose. The second one, accountability. It is very closely connected with the first one. The documentation needed in the GLP complaint conduct of a study will tell years later who was doing what and who could be held accountable for these mistakes. That's what accountability is required. They were performing because previously, if you see, if you go back, the human being, when they started their life journey first into the world, how they used to go for fire, two stone pieces they used to uh, right eh, then that spark used to come, and that was uh, the method. Today you are having different types of lighter where you are getting spark and you are getting fire. So somewhere this information came in different text, different language, different era, and today we know that human being used to be like that in past days. But that's what accountability or the record of the performance, you will able to know that years later who was doing what, who could held accountable for the mistake. If any mistake has been done, if the mistake was going on for the long time, who were the people involved in the which department is responsible for that, that you will come to know. The third one, GLP increase awareness. It awareness of management for the never ending task to strive for optimal quality and transparency of the studies conducted at their test facilities of study directors for the orderly performance of the studies they are to control. So it will increase the awareness of the management, right? Quality and transparency of the studies, optimal quality. We can improve if you have somewhere, if you know the this procedure we were doing till now, so it was somewhat not a healthy one. So we can rectify where is the problem, which step we can change so that we will get some more high quality, like that. Scope of good laboratory practice. So that the scope of good laboratory practice as it is defined in OECD principles, and which states that GLP encompasses the non-clinical safety testing of test items contained in the pharmaceutical products. Non-clinical safety testing is the sense what? Non-clinical or preclinical animals we are using. At the same time, we are expecting the data which will helpful during the clinical safety. That's what we do in preclinical. We are using rat, mice, pigs, rabbits, different which body, animal body is resemble to the human body. That model we are selecting, and we are testing the drug. So what can be done for safety testing of the test items? The pesticide products, not only pharmaceutical products, cosmetic products, veterinary drugs, as well as food additives, feed additives. Here two things are there: food additives and feed additives. Both are different, right? Feeding to the animals. Industrial chemicals, so many industrial chemicals it is there, which also need to be non-clinical safety testing. Forget about the whenever you will use the what you are going to have the data for that safety purpose. You might be aware that this is safe. This is safe. For example, Dettol. When you are purchasing the Dettol from market, you know that if Dettol will fall down in your hand, nothing is going to harm you. How you know? Because you are using it for since long time, and you don't have any fear towards the Dettol. But at the same time, if any strong HCl bottle will be there in your hand, how safely you will handle? Because you know that ki HCl will, if it will fall down in my hand, immediately it will burn my skin and I will get the wound. So that's what somebody tested, some lab tested ki this much concentration of acid is harmful, this much concentration of alcohol is safe for the human body. We know. So that's what non-clinical safety testing. It is very, very important. These test items are frequently synthetic chemicals, but may be of natural or biological origin, and in some circumstances may be living organisms. Yes or no? The purpose of testing these test items is to obtain data on their properties or their safety and respect to human health or environment. These two was very important. We are not only bothering about the human peoples or the person involved in that lab on that particular day on that particular procedure, or that particular surrounding, the area, the district, the city, the town, even to the environment. Because since it is lab and we are expecting lot of wastage, when the lot of wastage comes out, definitely there may be the toxic substances, and it is going to be again going to release into the environment. That has to be prevent. The areas of application, that is also very important thing. Here we need to discuss that the good laboratory practice is applicable to safety studies in two major areas. What are those two major areas? The first one, effects on human health, and the second one, effects on environment. That's what it is required, no? Otherwise all the lab they can design their own guidelines and they can do their own. Government need not to interfere in that. The governing body need not to interfere in that. Why the governing body is giving the rules and regulations? Ki if you want to produce so and so product, you need to follow this procedure or this method or this chemical or this raw material. There is a guidelines. Why? What the benefit government will get? What the benefit the governing body will get? Because it is concerned to the public health. It is concerned to the environmental safety. So these two areas may share some types of studies that have to be concluded in order to test the safety of the respective test item, but their test the study types may exclusively be required for one or other area, right? The second point to be covered in the judgment on the necessity of GLP adherence, these studies are not only conducted to obtain the data on properties, but also safety, which is intended for the submission to appropriate regulatory authorities. So here we are not only concerned about the what are the data we are supposed to have and supposed to record. At the same time, during that method, during that procedure, it should not harm machine, product, human being, environment. Everything we need to consider, and by considering all these things, how effectively we can design our method, we can design our tactics. That is the point. So here next one, human health and environmental safety. May any guidelines, first thing they are going to consider that human health and environmental safety. Forget about what the product you are preparing, what product you are launching, what kind of dosage form you are going where you have planned, what kind of the things it is going to come out from your laboratory. You forget all those things. You need to have to consider that human health and the environmental safety. The laboratory testing that is performed in a non-safety related way, non-safety related way. Uh, okay. Here one example given. You can see in this physical chemical parameters of pharmaceutical chemical substance, or it is also called physicochemical. Let me use the term physicochemical parameter. So everything, it's a solubility, refractive index, optical rotation, or density, or extractive value, ash value, uh, whether it is acidic, everything might not be considered as related to the assessment of human health risk. For example, pesticides or other environmental substance, however, parameters like their vapor pressure or their acid base equilibrium constant, or their water octanol partition coefficient, all of which are will determine the nature and extent of the environmental dispersion. The accumulation will certainly be safety related data that have to be generated under GLP. So scientifically we need to understand it is not that somewhere if it has written then it's okay. For example, now we are very much aware about alcohol, we are very much aware about all the non-polar organic solvent or polar organic solvent, we are very much aware, but which chemical which we don't know and which you are going to produce or manufacture in your lab, what is the outcome of that chemical? If it is harming the small insect also, which it is not supposed to, it has to be mentioned by the lab, by the products, and by the body. The data to be generated under GLP have to have a connection to the assessment of safety for their two field of human health and for the environment. In this regard, there are apparent differences between these two fields. The necessity to follow the strict regulations of GLP will be determined by the two basic principles of scope of GLP. The first one, the safety relatedness of the investigation and the forcible submission to the regulatory authority, the interpretation of which may, however, under different circumstances, lead to different answers and the conclusions. So here I am concluding the class. Tomorrow we'll continue.